Welcome to the exam marking video for the nervous system. Now before we do this exam marking video, this is a summary of the key things you need to know that pretty much all the questions link to. Now whenever we have a question about the nervous system, we need to understand there are two types of responses. The first is a reflex action, which is automatic, it's rapid, and it happens without thinking. And the second type of response is a coordinated response, which is a response that we have to think about. So a reflex might happen when you touch something hot. You don't have to think about it, your hand just moves away. A coordinated response involves thinking. For example, you smell some nice food and you decide to go and walk into the restaurant you've just smelt the food from because you've thought about it and decided, yep, I would like to go here. So there's a difference between reflex, automatic actions, and coordinated, very thought through responses. We need to know the details of a reflex action. You have a stimulus, which is a change in the environment. That stimulus is detected by the receptor. That's the first thing receptors do. And the second thing receptors do is to generate an electrical impulse. And that impulse is carried first by a sensory neuron, which goes all the way to a relay neuron, which is usually found in the spinal cord or the central nervous system. So it could be the brain as well, but it's usually the spinal cord. This then passes the impulse onto a motor neuron, which then controls an effector, such as a muscle which is able to contract, causing a response. Between the sensory neuron and the relay neuron, and between the relay neuron and the motor neuron, we have a synapse as well. So if you know and understand all the details amongst those things, then you'll be very well prepared to answer exam questions on this topic. So if we get started, we can straight away identify all the different parts that we've just talked about on this diagram, which shows a reflex arc involving the arm moving if the hand touches something hot. So we know that if we have something that's touching something hot, first you're going to have the receptor, which detects the change from the stimulus. This information is carried by a sensory neuron, which then passes the information across a synapse to a relay neuron, which then passes the information to a motor neuron that connects to an effector such as a muscle, which is then able to contract, and that's in the arm, so your hand will move away from the hot object. And so that nicely helps you to work out what the answers to these questions will be. Receptor is A, effector is F, sensory neuron is B, and the synapse is D. There are some clues here that I've written that might help you to understand exactly how you work out what is what. Usually you can work it out. For example, the effector, you know, is always usually a muscle and is always connected to a motor neuron. So you can use those two clues to work out where the effector is in the diagram. For the sensory neuron, you know it's between the receptor and the relay neuron, and for the synapse, it's always a gap between two neurons. Part C contains DNA. Which part of the nerve cell is C? Now, I already know from this list that only one thing contains DNA that we know about, and that is the nucleus. So that is the organelle or the subcellular structure that I'm going to tick. 1F. Reflex actions are automatic, as we said at the start of the video, and do not involve thinking. Why is this an advantage? Well, if you touch something very hot and you have to think, let me move my hand away, by the time you make that, you thought that through, you would have burnt your fingers. So we want to make reactions faster to protect us, to keep us safe, to avoid getting hurt. Those would all get you the mark there. Two factors for the next question that make you slower to react to a stimulus. Well, we know that alcohol slows us down, which is why drink driving is illegal, of course. Caffeine actually speeds us up, and actually reaction times improve with caffeine. And daylight, if things are more visible, it's probably not going to slow us down. In fact, it might help us see better and therefore react more quickly compared to the night time. Practice might actually help us speed up with something if we practice our reaction times, but tiredness of course slows us down. So again, drivers for example, because we're slower to react, are advised not to drive when tired. Question 2. This question is about the nervous system. A ball is thrown towards a boy. As the ball is thrown, information passes along the pathway to allow the boy to catch the ball. Draw one line from each action to the correct part of the pathway. So again, we've got the same thing we talked about at the start, and this is the correct answer. So retina cells in the eye detect. Remember, we know that receptors detect the light, which is the stimulus, from the ball. So the answer here would be, it's talking about retina cells which do the detecting of the stimulus, so our line should be to receptor. The impulse that reaches the brain which sees the ball and sends an impulse to an arm muscle. So we know that coordinators, which is something we hadn't talked about at the start, but I mentioned coordinated responses, we must think about what to do. So we receive information and then we send an impulse. So it does two things. It gets information in, it makes some sort of decision, and then it sends the right signal out so that we catch the ball properly. So that is the coordinator. Muscles we know are effectors, and we know that this is the response because that's the outcome. Students in a college made this hypothesis. Reaction time will increase as the time you have been awake increases. So in other words, what they're saying here is reaction times get slower the longer you are awake, probably because you're more tired. This is the method that is used. So let's have a look at the method. So first they said find 5 volunteers willing to stay awake for 24 hours. Now I imagine that's to become tired, and so we can investigate our hypothesis. Immediately I'm thinking 5 people in an experiment, probably not a lot of volunteers. Is that quite a small number? 2. Keep the volunteers in a room where they can study, use an exercise bike or watch TV as they wish. Now it says as they wish, but what if different people do different things? Something about control variables I'm thinking about here. They're not really controlling these things. Provide water, food, coffee and tea. I'm already thinking, coffee, caffeine. Interesting. Because that's going to help people feel more awake. And if they're not controlling that, could that be an issue? Then they're measuring the volunteers' reaction time every 4 hours using a computer program. I'm thinking, computer programs, they're quite accurate, that might be good. What is the independent variable in this investigation? The thing that has been changed. Remember, chimed. We change the independent variable and we measure the dependent variable. So the independent variable is the time awake. Different people will be awake for different amounts of hours, or every 4 hours is going to be measured. The students used a computer program to test reaction time. What other method could we use? So we've done this in class. We've done the ruler drop test. So here is a good method. Remember, good methods usually start with some sort of imperative verb to make them really nice and concise, and we make sure we're being as specific as we can. So we place a ruler in line with the person's first finger and thumb, vertically starting at 0 centimeters. So these are all control variables that help me keep consistent in my method. And by specifying how they hold it is important. So if you wrote something slightly different, as long as you wrote how they're going to hold the ruler, that's what gets you the first mark. Then drop the ruler at an unpredictable time, because we want them to react to it. Then the distance the ruler drops in centimeters is recorded and converted into a reaction time using a chart. So that's important because the third mark is for specifying how you are going to use the information, which is the centimeters, the data is centimeters, and convert that into a reaction time, because that's what we want to measure. And another control variable, if you didn't talk about holding it from your finger and thumb and starting at 0 centimeters, you could have said control the amount of light in the room, because obviously if it's dark it's harder to see, maybe harder to react. So that's there in case you didn't talk about the finger and the thumb and the vertically at 0 centimeters. Next question, computer program, why is it an advantage? So the advantage of using a computer test is that it's more accurate, or you can say it's more repeatable. Remember, repeatable means when you do it again and again, you get the same results when you repeat the investigation. So if I was to do it on the computer again and again, then I would get very similar results, whereas if I use the ruler, I might find my results vary more. So it's less repeatable to use the ruler drop method and more repeatable to use the computer. It's also more accurate because it's more, the definition of accurate is it's close to the true value. So I have a particular reaction time, and the computer is going to be closer to my true reaction time than the ruler drop test might be. Okay, so here we have data, and we know that the results are as follows. So every 4 hours they did the reaction time, they measured the reaction time, and they found a mean of the 5 volunteers each time. But there's one missing which we need to find, so we simply add up the values, divide it by 5 because there are 1, 2, 3, 4, 5 numbers, and you should get 0.556, or you can round that to 0.56 seconds. So you get 1 mark for showing you're working out, and 1 mark for having the final answer there. You're then asked to describe the pattern of results for the mean reaction time as the time awake increases. In other words, as the time increases here, what is happening here? So if we have a look, it seems to go down at first, but then it's roughly the same, but then after 12 hours it seems to be, these are quite similar to each other, but then after 12 hours it seems to be somewhat different, much higher. So there's a couple of things you can say to show that. First, as the time awake increases, the reaction time stays roughly the same. Or you could say dips slightly. That's the first mark. But then you need to say, however, it increases from 12 hours awake. So after 12 hours, it seems to be much higher than before. Those are your 2 marks. You have to use data in this way because it says it's 2 marks and we need 2 clear points. Do these results support the student's hypothesis that reaction time will increase as the time you have been awake increases? Well, you could say yes and you could say no, as long as your justification matches what you are saying. So if you said yes, then you need to say yes, reaction time will increase as the time you have been awake increases, because overall reaction time goes up. When you're awake for 4 hours, then it's a very quick reaction time, but when you're awake for 24 hours, it's much slower. Or you could say no, because it stays the same at first. So it's not fair to say that the longer you are awake, the longer your reaction time, because they're pretty similar between 0 to 8 hours. And so you need to say no, because it stays the same at first and only goes up after 12 hours. Or you could say it goes down slightly, in fact. As long as your answer matches your reason, that's fine. So, 2 ways to make the investigation more valid. So we need to give 2 ways to make it more valid. Well, first we could use more volunteers, because the more volunteers we have, the larger the sample sizes of our investigation, so the more valid our results will be, or the more valid the investigation will be. The second thing is to make sure that they all do the same activities, so at the same time. Or in other words, you give them the same food or coffee at the same time, because remember, in their experiment they said they can have coffee, tea, food, exercise as they wish. So it's not the same, it's not a control variable. But the key thing about being valid is your control variables have to be all kept in place. So make sure they do the same activities at the same time, or they have the same food or the same amount of coffee at the same time. Those are control variables, and if they're kept in place, that would make your investigation more valid. So the more valid the more valid investigations tend to have all the control variables in place and their data is really reliable. One of the ways of making data reliable is to use a large number of volunteers or a large sample size. Okay, now we're looking at a question on synapses, which is the gap between between two neurons we talked about earlier. So the diagram shows how a nerve impulse passing along a relay neuron causes an impulse to be sent along another type of neuron. So you've got impulse going along relay neuron, and then that impulse is passed on to this other neuron. What type of neuron is this? Now we know that the order goes sensory neuron, then there's a gap, and there's a synapse. We have our relay neuron, there's another synapse, and then there's always a motor neuron. And we know that the gap labeled Y is called a synapse. Describe how information passes from the relay neuron to neuron X. In other words, what happens at a synapse when we go from a relay to a motor neuron. You can use the diagram to help you. So, first of all, a chemical is released. If you said neurotransmitter, great. The chemical diffuses, or you can even use the word crosses, but it's better to say diffuses. I would stick to this word here. Diffuses across the synapse. Again, you could say gap if you forgot what that word was, but it's best to use the technical language. Diffuses across the synapse, and then you can say the neurotransmitter or the chemical binds, that's probably the best word to use, or attaches to the next neuron, and that causes an impulse there. Those are what the 3 marks are for. Right, next. Scientists investigated the effects of 2 toxins on the way in which information passes across synapses. So the toxins are affecting the synapses and the chemicals crossing the synapses. Here are the results. So the first toxin, curare, decreases the effect of the chemical on neuron X. So in other words, even though the neurotransmitter is going across, its effects are reduced. So that means there'll be fewer impulses generated here if curare is, the toxin called curare is present. If there are fewer impulses here, the question is saying, describe the effect the toxins have on the response by the muscles. So if the motor neuron is producing less signals because curare is decreasing the effect of the chemical on the neuron, then this will decrease the muscle contraction. So the tricky thing here is realizing that the toxin is affecting the synapse, but the question is about the muscles. So your answer must be in terms of what happens to the muscle contraction. For strychnine, it increases the amount of chemical made in the relay neuron. So if there's more chemical made here, then more will diffuse across. So will there be more or fewer impulses? I think if there's more chemical, there's going to be more impulses, and if there's more impulses in the motor neuron, then what's going to happen to the muscles? They're going to contract more. So that's the mark there. So to summarize, if there's a smaller chemical effect on X, if we're talking about curare, there will be fewer impulses, so there will be fewer muscle contractions. For this toxin, if there is more chemical in the relay neuron, more will bind to the motor neuron, there will be more impulses, and there will be more contraction.